Here we go. As you recall, last week I was discussing the function of genes. And I was discussing the... this thing is so irritating up here. I was discussing the function of genes. And I was discussing how genes work, what they do, what they are. And when I talk about this kind of stuff, you should try and put this in the context of genetic engineering. When I talk about what a gene is, when I talk about what genes do, when I talk about the functions of genes, think about it in the context that you're debating people about the use of this technology. And as I said 3 weeks ago when I started this class, it's not about opinions, it's not about beliefs, it's not about waving magic wands, it's about science, good science, and useful science, science that can get to the truth from a scientific point of view, and using all of those facts in order to make opinions. And I was absolutely ecstatic today that President Obama had in his inauguration speech that we're going to get back, as he said, to using science to make good policy decisions, something that hasn't been done for the last 8 years. And so it's important when you think about genetically engineered plants and animals and the technology we talk about, I want you to think about what a gene is. I want you to think about what genes do. I want you to think about how they function. And I want you to think about the things that I've been trying to teach you because those are the things that are necessary to form logical and informed opinions. And so last time I talked about what are genes. We talked about the evidence for DNA being the genetic material. I talked about these remarkable experiments by Griffith, as you recall, who invented this word transformation and transforming principle working in this black box. I talked about Avery. I talked about Avery, MacLeod, and McCarthy and their remarkable experiments uncovering that DNA was indeed the transforming principle. We talked about how the glowfish experiments were really an extension of the things that Griffith did. And we talked about antibiotic resistance being transformed, and a lot of things that we've discussed, trying to put it together more in a conceptual way. And so I want to continue on this theme where I left off. What are genes and how do they function? And today what I want to do is I want to talk about a conceptualized gene. I want to talk a little bit about how genes are turned on and turned off. I want to talk a little bit about how genes are replicated. And then we're going to have some applications of some remarkable technology, talk about mutations and how they occur, talk about differences between gene activity in prokaryotes and eukaryotes, and then hopefully I'll have time to wrap it all together and begin to be able to conceptualize for you and hopefully have a little bit of a discussion of what this means in the context of a debate about genetically engineered organisms, humans, plants, animals, fungi, bacteria, and how all of this knowledge that I'm trying to transmit to you imparts your ability to engage in the debate, engage in the discussion about this new revolutionary technology. And so let me begin by reminding you of what we talked about. Can you hear me, Dora?
Yeah.
Okay. Let me remind you about the conceptualized gene. Remember that genes, I said last time, lie on chromosomes, and genes have specific positions on chromosomes. And genes, as I'll reiterate over and over and over today, genes are discrete units. Discrete units along the length of a continuous DNA molecule. And remember I said last week that the molecules in your chromosome, remember, Derek, I said they're linear. And remember I said the molecules in the bacterial chromosomes are circular or round. And so we have this idealized gene, which I love talking about because it has a beginning. And remember I said the beginning was the 5' end. It has an end. And remember I said the end was the 3' end. The beginning has a switch, as you've learned in discussion, that turns the gene on, turns the gene off. And of course we have an end. It is these particular features that define where the gene is, Max, on the chromosome and how it works. And what I want to emphasize here, and emphasize it absolutely emphatically, Kathy, is nothing occurs without specific sequences. That's what guides everything in biology. And those specific sequences, I said last week, are sequences made up of DNA, and we talked about the structure of DNA. So ultimately, it's chemistry and the ordering of particular molecules that leads to all these great things that happen inside of a cell. And so a gene has a beginning because there are specific sequences at the beginning of a gene. A gene has an end because it has specific sequences at the end. And the gene is unique because the order of nucleotides in between is unique for that gene. A genome is unique because the order of nucleotides is unique for that genome, and so on and so forth. And so now let me go through for you the anatomy of a gene so that you can understand in conceptual sense how all of these things work. And here's Bob Goldberg's bird's eye view of the anatomy of a gene. We have the beginning and we have the end. We have the beginning, we have the end. We have two strands on the DNA molecule in a double helix. Only one strand, as I think I've said last week, but I'm going to say it again, only one strand of the DNA makes sense in terms of directing the cell to make a particular protein. The other strand is just there for structure because the DNA is double helix. It has to be there, and to pass the information on from generation to generation. And so we have two strands of the DNA. One is called the sense strand, one is called the transcribed strand, sometimes the antisense strand. The genetic code, which you learned about in discussion last week, is the sense strand. The genetic code is the sense strand. The codons, the nucleotides, which direct which amino acids to be in a protein, are in the sense strand. And that sense strand goes from the beginning of the gene, which, as I've said, is called 5', to the end of the gene, which is called 3'. But the direction of the gene is always specified in the form, Nancy, of the sense strand. A gene is always defined in terms of the sense strand. And when you did your quiz for last week and you did this blast and you came up with this gorilla and you came up with a human SRY, you were blasting against sense strands of genes, essentially. You were looking at the strand that made sense. And that's why you could come up with what the protein was. And so we have the sense strand and the transcribed strand. Sense strand goes 5' to 3. Remember your double helix. The double helix is anti-parallel, 5' to 3, 3' to 5. That's very important conceptually. Don't ever forget that, or you're going to get your sequences always wrong, particularly on your take-home exam, which will be handed out next week, and your oral exam about 2.5, 3 weeks from now. Okay, the beginning of a gene then is defined by what we'll call the switch or the promoter. And notice that there's a particular sequence here. And notice that the sequence on the Watson strand is different than the sequence than on the Crick strand. So notice we have 5', TATAAT. That's a specific sequence. Let's just say that the switch of most genes has that particular sequence. And so this sequence marks the beginning of a gene. And the cell can recognize the beginning of the gene because it can recognize that sequence. Notice that that sequence is different than what's in the gene itself. Notice that that sequence is different than what's at the end of the gene. Everything is by sequence. The beginning is a specific sequence. The gene is a specific sequence. The end is a specific sequence. That's how everything works inside the cell. And so notice here that there's a particular sequence here, but notice that the sequence of this strand is different than the sequence of that strand. And that's extremely important in a sense, for reasons that I'll tell you in a second. Now notice that at the end, there's also a different sequence. And that's called the off switch or the termination switch. And so again, the beginning has a specific sequence. The end has a specific sequence. And Rose, if you were looking at the whole genome, simplistically, of a genome whose sequence you just determined, Do you think you'd be able to use sophisticated computer programs to figure out where genes lie in this mass of nucleotides that you just sequenced?
Assumably, yes, because you could look for the specific termination and start sequences.
Yeah, exactly. Do you agree with that, Dora?
Yeah.
So from a simplistic point of view, if you just had an ocean of DNA sequences, let's say you sequence some incredible genome, you'd be able to write some sophisticated coder programs that would be able to say, here's this gene, this gene, this gene, this gene, very simplistically, of course, on the basis of those signature sequences. And then simplistically, Rose, you might be able to say, let's just ignore introns for a second, that you might be able to look between these sequences and see there's a stretch of nucleotides that makes sense from a genetic code point of view, that is code for a protein.
Yeah.
And don't you think you might be able to have, Lauren, a very specific program that might be able to say, here's the beginning of a gene, here's the end of the gene, and now, using the genetic code, here's what the gene codes for.
Yep.
And that's exactly what we do, and that's genomics. And that's what you did using the BLAST program that you used to analyze and find out that it was the SRY gene. And so one can use these sequences as signatures in order to be able to basically say where the beginning of a gene is, where the end of the gene is, and what's in the middle. Okay, now listen carefully to me, Anna, and Mikhail, and all of you, because here's where it gets a little bit technical. Notice that I said that the sense part of the gene is this 5' to N3' strand. So what is this nonsense or transcribed strand doing? Well, this nonsense or transcribed strand is, in fact, the strand in which the RNA polymerase molecule, which is responsible for putting together the messenger RNA, travels down and uses as a template. Because remember, everything in genetics is complementary. The messenger RNA is complementary to the gene, but only one strand of the gene. So notice, let's come back and say, here's the beginning of the gene, here's the end, and let's say that gene has to be transcribed. Notice that at this start switch, this enzyme RNA polymerase, which you talked a little bit about in discussion last week, knows to bind, that is an enzyme that binds to the DNA at this specific sequence. But now the polymerase has sort of a problem or a dilemma, Chris. It doesn't know which strand to be able to transcribe. It's only going to transcribe one and in a particular direction. And the way it knows that is by the difference in the sequence of this particular start site. Because it will notice that there's this sequence here, and therefore it's supposed to transcribe this strand, as you can see, the transcribed strand. And so the polymerase binds, yes, to the switch or the promoter, but by the sequence difference of the 5' to 3' and the 3' to 5' strand, it knows which strand to transcribe. And in a gene, the strand that's transcribed is always 3' to 5. Always the 3' to 5' strand. Always transcribed, never 5' to 3. And the reason for that, as you can see in this diagram, is that the messenger RNA that's made is complementary to the transcribed strand. And notice, Ben, that this little blue arrow going 5' to 3' and this red arrow going 5' to 3' is a double helix. It's essentially a double helix. Everything is a double helix, whether it's DNA pairing with DNA or DNA pairing with RNA. And the only way the gene knows to make the specific RNA is that the polymerase moves down this gene, transcribes this transcribed strand, puts a complementary A here, complementary G, complementary C. It puts a U in because there's no T in RNA, and we'll talk about that a little bit later. Puts a C in, and so forth and so on. Again, the gene has a beginning, the gene has an end. They're marked by sequences. And the sequence difference in the switch between the Watson and the Crick strand tells what strand is transcribed. And the transcribed strand is always going relative to the gene 5' to 3', because the message is always made 5' to 3'. And notice that the sequence of the message is exactly the same as the sense strand. AGCTCGAAC, AGCUCGAAC, exactly the same. And that's exactly why the sense strand is called the sense strand, because that's where the genetic code is, because the messenger RNA, as you know, will be translated in the cytoplasm to make a particular protein. In addition to the gene, keep in mind that the messenger RNA has a beginning and an end, and those were those signals that you talked about in discussion last week, and we'll talk about those in a few minutes when I talk about genetic code. But clearly, there's an end. Notice the P, 5'. There's an end, 3', OH, 5' to 3' direction. Exactly the things that I've told you in previous times. And so, in a sense, that's the anatomy of the gene. Now, what's upstream? Well, this is a continuous chromosome. So there's going to be genes running all the way to here. And then what's downstream is that there's going to be genes running all the way down here. And they're all going to be individual units along the chromosome, having starts and stop switches and being transcribed in different ways. And so I'm not going to go over this because you can read it, but clearly this is the simple gene. This is the anatomy of the gene. But what I just said was, I think the most important thing, Zach, is that we have this chromosome, and there it is. Brian, is this a chromosome before or after DNA replication? Is this chromosome, this picture of the chromosome, is it before or after DNA is replicated? Think about the process of cell division in a plant or an animal. Is that before or after DNA replication? What's your hypothesis?
Before.
Before. Jessica, do you agree? Jessica agrees. Derek, what do you think? You disagree. Before or after DNA replication? Dora, what do you think?
I agree that it's before.
It's before. Lauren? We have before, after. Taylor? After. Okay. Why after, Taylor? Take the microphone. You're guessing? Well, what has to happen when a cell divides?
It splits in two.
What? I can't hear you.
It splits in two.
What has to happen to the chromosome, Hiromi? I can't hear you.
It has to split.
Do you agree, Dora?
Yeah.
You want to change your mind? Is this before or after DNA replicates?
Oh, me?
Yeah.
Oh. What? Isn't it before? It hasn't.
Still
split yet.
still haven't changed your mind. Okay. Lauren, can you explain why you think it's after? I mean, yeah, before? Sorry.
During cell replication, the chromosome has to split in two, so there has to be two copies of the DNA
Before
in one chromosome.
cell division, the chromosome has to split in two, so there's got to be two parts of the chromosome to go into each cell as it divides. So normally, before cell division, it would just be the top part, and then after the DNA synthesis, the bottom part appears. So in a sense, in this chromosome, we have two double helical molecules, and both of them are going from one end to the other. And notice that this little string comes from one of them, and it's a long continuum. And notice, as I said, gene 1, gene 2, gene 3. Notice that we have gene 1, gene 2, gene 3, start, end, start, end, start, end, specific coding sequences, mRNA sequence, and protein sequence. This is a very important concept, enormously important, because it says, Marvin, that the sequence on the gene specifies the sequence of messenger RNAs, specifies the sequence of amino acids in the protein, which specifies 3 different functions. Everything because of sequences, complementary bases, and relationship between DNA and RNA and protein via the genetic code. And so notice that the position of genes 1, 2, and 3 in the chromosome, each gene has unique order of sequences, and you can see that here. AGCTGGT, CCAGCT, CGTAA. Different sequence, different protein, because different order of nucleotides, different genetic code. And you can see that all of these genes function as a unit independently of another gene. Tiger, can you come to the board? Doro, you want to go to the board in Davis? Or Faria, you guys want to go to the board? Can you see what's going on down here? Use this. You have a little whiteboard there. Design an experiment to show that genes work independently of each other using things that we've discussed over and over and over in this class, particularly the first day. Design an experiment to show that genes work independently of each other. So using genetic engineering or anything that you want, we've given many, many examples of what you can do using genetic engineering. So, Tiger, start off. What would be your favorite gene to show that it works independently of other genes? That is, other genes don't influence its activity in any way. Yeah, pick anything that we've talked about in class relative to genetic engineering. Hint, hint. Okay, antibiotic resistance. Do you agree, Dora and Faria? Put your antibiotic resistance gene up there. Just draw it out. And I think we need a microphone that works, Daisy. It's getting old with these microphones. Works, got to talk into it.
Antibiotic resistance.
Okay, so put your antibiotic resistance gene up there. And you just draw it out, you know. A gene circular? Is it a circle gene?
It could be.
You're working with the chromosome or you're working with the gene? So there, you're working with a naked gene or you're working with a circle? What do you guys, Dora, what are you working with? The gene or the plasmid?
The gene.
Now, thank you. Just draw the gene. I'm not aware of a circular gene. Are you? That's a funny-looking DNA molecule. Looks like a fish. I never saw a DNA molecule look like that. Where's your DNA molecule, Tiger?
It's just this little part of the chromosome.
Is that what you're working with in your lab, that part of the chromosome? Is that what we've been talking about for the last 3 weeks in discussion and in lecture? What are we working with?
I guess a piece of DNA.
Can you talk louder with the microphone?
A piece of DNA.
Ah, now you're working with a piece of DNA. Exactly. So we got a piece of DNA. Do you agree, Dora?
Yeah.
Yeah, you sure?
Yeah.
Where's your double helix? So that's your antibiotic resistance. Now, what's the prediction? What's the prediction? What is the prediction, Zach? If you can hand Zach the microphone. What is the prediction if that gene works independently of other genes when you stick it back into, let's say, a bacterial cell that's antibiotic sensitive?
It should give that trait to whatever you stick it in.
Give that trait to whatever you've, what? Stuck it in? Sounds
If you transformed
gross.
it into a bacteria for
Huh? You ever stick anything in anything? What?
If you transformed
You're turning
it
red.
into
Sometimes it's a lot of fun. In any case, let's not go there. In any case, when you stick it in something,
if you
in a bacteria, right? It should, God, are you red? He's red as a beet. Can you see it, Dora? In any case, what's your
the bacteria
prediction?
should present the trait that gene codes for.
No, I said doesn't influence any other genes, only works independently. What's your prediction?
It should function the same way it does when all the other genes are still with it.
Give me a specific prediction.
It should present the
Help him out, Elaine.
The bacteria should function as it normally does.
Thank you. The bacteria should function as it normally does. Because if you stuck this gene in the bacteria and it influenced all the other genes, do you agree, Faria? The bacteria is going to croak.
Yeah.
Or it ain't going to look the same way. Right? So what happens, Faria, when you stick that gene in an antibiotic-sensitive bacterial cell? Can you show me? Draw a cell and stick it in. Go on. Yeah, good. I never saw a round bacterial cell. I've seen round eggs, but I've never seen round bacterial cells.
What shape do they usually look
Well,
like?
we've shown it on the, I know you're spending the whole weekend studying your handouts, so, and the book and everything else.
Yeah, move off to the side a little bit.
Our oral exam in this class is going to be exactly like this, except everything that you put on the board you're going to have to explain. If you say DNA, you tell me the structure of the double helix. If you say double helix, you tell me about nucleotides. If I ask about nucleotides, you tell me about carbon, hydrogen, nitrogen, and oxygen. If you say transformation, you tell me about transformation. We go from one thing to the next. It's fun, truthfully. It is, and you learn a lot. Okay, Faria, you put it up there. Is that cell antibiotic sensitive or is it resistant?
Resistant?
It's resistant. And who did that experiment first? What were the names of the people that did that experiment first? Is this sensitive or resistant?
Resistant.
Can you put it down? You still have a plasmid here. I thought you started with this DNA. I don't see the plasmid. Okay. Is it resistant or sensitive?
It's resistant.
Yeah. Tell me why it's resistant, Faria. Show me how the cell becomes resistant. What does that piece of DNA do once it gets in the cell relative to what I'm talking about today? Albert, don't smile because you may be in the same situation. I'm teasing.
Whatever the gene codes for, it'll be expressed in the gene.
How? Take me through the process simply. Does the gene get transcribed into protein?
Yeah.
It does? The gene is transcribed directly into protein?
Well, no, first into RNA.
Thank you. And then what happens? Does that antibiotic resistance gene, Tiger, have a specific sequence?
Yes.
Does the antibiotic resistant protein have a particular sequence?
Yes.
What's your hypothesis for how this antibiotic resistance gene can prevent this antibiotic from killing the cell?
I think that it'll.
You think? What's your hypothesis?
My hypothesis is that it will create specific proteins, probably enzymes, that will either help this, keep it from entering the cell wall, or will destroy it once it gets inside
Or
and keep
pump
it from
it out.
replicating.
Excellent. Do you agree with that, Dora?
Yeah.
Okay, great. Good job, guys. Under pressure. Good job. In any case, that's the kind of experiment that shows that genes work independently of each other. Now, the question really becomes, and I think one of the most exciting questions of biology, is how are genes turned on and turned off, let's say humans, from conception to death? And would you agree, Samantha, if we understood that, if we understood the switches and how they work from the time of conception to the time of death in every cell, that we know an awful lot about how we live?
I think we would know a lot.
And do you think that if we know all of that, however long that it takes, and we know when genes get switched on, when they get switched off, in the heart, the liver, the brain, what genes are active in the brain, what genes are active in the heart, what genes get active when we get old and senesce, what genes are responsible for illness, fighting illness, aging, everything. Do you think once we know all of that, it may lead to immortality? Because look at the technology that we're using today. This is primitive.
I think that's a scary answer.
I didn't say it was scary or not. That's a value judgment. I said, do you think it's possible?
I truly don't know.
What do you think, Maura?
I think it would have the potential to do that.
Have the potential. Do you think that we as human beings, from a genetic point of view, are a closed system? Do you think we have an infinite number of genes in our chromosomes? Do you think we have a set number of genes in our chromosomes?
Yes.
Do you think we can know the functions of all those genes?
Yes.
Do Do you think we can know how those genes get turned on and turned off in a sequence of events at some point in time in the future? We don't know it now.
I think we
Do you
could
think
get
we
there.
could understand, once that sperm goes right into that egg, exactly what happens and how genes are switched on to make a little human embryo, and how that embryo develops into this beautiful little baby by making all these different organs and stuff, and what time it occurs, and what proteins are made, how the proteins interact with each other, and how these organs get established over a time sequence of events?
I think that at the rate we're going, we definitely have.
And
that
from what you know about genetic engineering, if you know that, think about the fly. That's primitive. You think that everything can become possible that's possible. And as you remember, think about that dead mouse. That dead mouse that was just cloned. In fact, there was another report of a dead bull that had been dead for 47 years, and they revived its tissues and cloned that bull into a prized bull that's a genetically identical copy of one that lived 50 years ago. And so, as I said the first day of class, do you think that all of these technologies will converge at some point when you consider the fact that it was only 50 years ago that DNA double helical structure was solved?
Yes.
So I would submit, and we can debate this point, and we will at some point, that if you know how the blueprint of our genome is read out from conception to death, that you'll be able to intervene in a genetic way, let's say not to cause immortality, but certainly things like cancer, certainly which are diseases of our own genes, and certainly a lot of viral infections and things like that will become totally the thing of the past, because we'll be able to deal with it from a rational molecular point of view because we understand how these things work. Now, making another human being is a whole different story. I don't know why we would do that, but that's way down the line. But the point here is that, as you discussed in discussion last week, there are these switches for both prokaryotic genes and eukaryotic genes, and those switches are the beginning of the gene, but the beginning of the gene only says bind this polymerase which transcribes the gene. The polymerase doesn't really have a big brain. The polymerase knows to bind to the beginning of the gene, the promoter, but it doesn't know when. And there are other sequences in the gene, in the upstream region of the gene, that bind other proteins, activator proteins and or repressor proteins that interact with this complicated enzyme to make a structure, as you can see here, which tells the gene to be transcribed. In the case of a bacteria, I'll give you a simple example. There's this gene called beta-galactosidase, which you might have read about in your chapter in the book. It's an enzyme that is able to break down lactose into galactose and glucose. When a baby is born, a mammalian baby, and it's fed mother's milk, it's all lactose. The E. coli is growing in the intestine of this baby. How's it going to survive when it needs sugars and it doesn't see glucose, which it really needs, but it sees lactose? Well, these E. coli beta-galactosidase, let's say gene, has a promoter that binds the polymerase, but it also has something else that I'm not going to go into that binds another protein, and that protein can sense whether there's lactose or not in the environment. And when the baby starts drinking the milk, starts breastfeeding with the mother, lactose goes into the system, goes into the intestine, goes into the E. coli. The E. coli senses the lactose. That E. coli proteins senses these things called, let's say, transcription factors simplistically. It changes something and then interacts with the polymerase in a very simplistic way in order to turn on the gene which breaks down, makes the enzyme beta-galactosidase that breaks down lactose into glucose and galactose, and it's glucose which is used by the E. coli to make energy. Well, the same thing happens, as you know from discussion last week, the same thing happens with eukaryotic genes to tell the gene to be active in a heart, active in a kidney, active in a skin cell, whatever. And again, it's no hocus pocus. It's in the DNA sequence. And so in the upstream region of a gene, upstream from this promoter, which binds this polymerase, there are these little sequences, and they're all different sequences. And those sequences bind different proteins that tell the gene what to do. Tells the gene when to be active in development, tells the gene in time, tells the gene what thing to be active in, what cell type or organism in time, and it becomes a very complicated process. And so with respect to the eye gene, it would tell that eye gene to be active in a head during the development of the eye. In the case of the SRY gene, it would tell the gene to be active in the developing gonads in order to specify that the gonads should go in the direction of becoming a testis. And so the point here is that the polymerase, again, has no brain. It will bind to here, but it needs other proteins to tell it when and where to be active. And those are regulatory proteins that interact with this particular enzyme. And so therefore, as I've said many, many, many times, it's the sequence that specifies the biology. Therefore, as I'll show you in a second, the eye gene can be engineered by engineering these sequences to not be active in the eye, but be active in the leg. Or any gene can be engineered by simply using genetic engineering and replacing these sequences with other sequences. It's a very simple thing. We do it all the time. Kelly's just published a paper, had her name on a paper, in which she was able to identify which sequences in a plant gene are responsible for activating that gene at a particular time in the development of a plant. And so there are these sequences that tell genes when and where they're supposed to be active, and those genes can be used, those sequences can be used as raw materials to do what we want. And so clearly, the insulin gene has switches for the pancreas, whatever. The point that I'm trying to make here, and I'm going to make it again today over and over and over again, is that a gene is modular. Brian, you can take the coding sequence of a gene and mix it with the switches of another gene, and vice versa. And you can take multiple switches. Let's say a gene that's active in the head. You can now engineer that gene to be active in the head, the leg, the wing, the antenna, by just stitching together these stitches, because they all work independently. It's a really remarkable concept. And therefore, there's no limits to what we can do genetic engineering-wise. The point here is that you can clone these things by genetic engineering. You can use restriction enzymes to bring them together into chimeric genes, just like you learned the first week of discussion. And you can transform, analogous to Griffiths and Avery, different cells to get these things to do different things. It's simply a sequence. It doesn't mean anything else. If you have the sequence, if you have the code, you can tell a gene to do anything you want. Or for that matter, Hiromi, you can synthesize your own gene, because you don't even need to clone anything, as I'll show you in a few minutes. This is the excitement of this technology. And this is what's advanced this technology, because without genetic engineering and recombinant DNA, I think I'm going backwards, yeah. Without genetic engineering and recombinant DNA, how would we ever even known what the gene was like? Because in order to know what the gene was like, you had to clone it first and sequence it. In order to find out that there's these switches here, you had to be able to clone them, sequence them, and find out what they do. And so the point here is that recombinant DNA, as I said a few weeks ago, led to some remarkable, remarkable, remarkable breakthroughs. Again, as I showed you, this eye gene is normally active in the head because the switches are only head switches. The switches come on when the fly goes through embryogenesis in the parts of the larva of the fly that gives rise to the head. But when we take the coding sequence of the eye gene, that what specifies the eye protein, and when we stitch it together with leg switches, we can get the normal eye gene produced, normal eye produced, and eyes on the leg. So it's all about mixing and matching. Now, what about this eye gene? Well, most genes in genomes just do one thing. You know, they do their thing. Like the insulin gene, Zach, only codes for insulin, and you know, it's involved in sugar metabolism. But this eye gene is a very special gene. It's called a regulatory gene because it regulates the activity of other genes. In other words, everything is sort of interconnected with each other. The insulin gene works independently. Insulin genes transcribed, the insulin protein's made. Insulin causes a lot of physiological interactions, but not genetic interactions. But this eye gene is important because it actually tells this head to make an eye. Kathy, would you hypothesize that in order to make an eye, you need only one gene or thousands of genes to make a complex structure like an eye or a brain or a head?
Thousands of genes?
Yeah, thousands of genes. But what tells those thousands of genes only to be active in the head? That's the eye gene. Because the eye gene specifies the proteins I just talked about that interact with this polymerase. The eye gene makes one of these regulatory proteins. And so what happens is there it is, conceptually. And this is the hottest area of biology today. This is what Dr. Harada works on. This is what I work on. This is what many people work on across the globe. And that is we have the eye switch and the eye gene. But notice that eye protein will turn on gene 1, gene 2, gene 3, gene 4, all which have to be turned on in the larva, in the head, in order to make the eye. And all of these proteins are proteins that contribute to the structure of the eye. So going back, conceptually, the eye gene would contribute to the production of one of these proteins. And going back conceptually, the eye gene would contribute to one of these proteins that interacts with this enzyme and binds to these switches. And therefore, because many genes might have those particular kinds of sequences, meaning many genes are active in the head, therefore many head genes have head regulatory sequences, the eye protein can bind not just to one gene, but to hundreds or thousands of genes, and in a sense, what we call form a regulatory network. The eye gene makes the eye protein turns on all of these genes, makes all these proteins, and makes an eye on the head. But by using genetic engineering, we can switch out the eye switch with a leg switch. And now that leg switch makes a eye protein, but it only makes it in the leg. And genes which are normally off in the leg because they never see this eye protein to interact with this polymerase, now all of a sudden see this eye protein, and genes that are normally silent, protein 1, protein 2, protein 3, protein 4 are now made, and those proteins contribute to this massive structure. Eden.
Would it be possible to make a head then on the
Yes.
leg?
Theoretically, yes. The problem is the head is a very complicated, it contains many organs, right? It contains a brain, it contains eyes, it contains a mouth. So really that forms during embryogenesis. Dr. Harada's lab, though, in that vein, discovered a gene in plants along the same lines that you're talking about, but it's different for plants, in which you make that gene active in a leaf. You know that a leaf is a leaf, but when that gene becomes active in the leaf, it turns that leaf into little embryos, which are technically can become seeds. And so in a sense, theoretically, it's possible to go in that direction, but it's highly complicated because an eye is one organ and a head is a mass of organs. But theoretically, if you know the right sequences and you know the right genes and you put a whole bunch and maybe stack a few together, you can do that. Samantha.
So if we're able to grow an eye on a fly's leg, but the eye is only grown on the actual fly, could we take a human body part and grow it on another animal?
Probably not because of antibody-antigen reactions.
Okay.
Although people do use pig hearts and have used pig hearts as a replacement for a human heart.
Okay.
But there are rejection things due to the immune systems. On the other hand, how about using stem cells in which they have the potential to become organs, know what genes you can switch on at the right sequence, engineer the stem cells in order to go in the direction of a heart, and learn how to mass that heart together into a tissue and have a heart replacement.
Where do you grow this heart? Do you grow it inside the body? Or
It's not being grown now. In the future with stem cells, you would grow it in a laboratory in a Petri plate,
Okay.
essentially. But of course, that's a much more complicated problem because you're dealing with a very complicated organ with many tissues and many different parts. It's not like a single cell. Albert.
Is it possible to get like the eye to work? Because it's the eye's on the
You'd
leg
have
now.
to innervate it with the nerves, and that would be a very complex process. You have to send nerves out from the leg on the eye to the brain, which is where everything happens.
What if you built a brain on the leg?
Theoretically, that could be possible, but you know, it's a long time into the future. Again, we're talking about making, the eye is not as complicated as a brain. And if you made a brain on the leg, you're going to have to send all the neurons out everywhere. And so it's not a simple process. Faria, do you have any questions?
I'm good.
Comments? Dora?
No comments.
Okay, let me go back and review how genes work, because I want to talk about a particular process. So remind you that the function of genes is to replicate and pass down this genotype generation after generation. Humans make humans, skin cells make human cells, bacteria cells make bacteria cells, and maintain the phenotype because the genes are replicated, you know, just faithfully. And so I've been discussing these genes, and I've been discussing in general this process in general, but what I want to turn to is the process that is responsible for all of us being here today, the process that's responsible for us proliferating into the future human beings, and that is this process right here of DNA replication. Every time you take a shower and you rub your skin cells off and the skin cells grow back, you have to replicate the DNA faithfully and identically to what it is, and you have to get it in a completely faithful way with very few mutations, although you know that there can be mutations because all of us have freckles and all kinds of other things, and you can get skin cancers, God forbid, so don't go into the sun because those are the results of somatic mutations, and you can get all kinds of things. But in general, as we said, you have to faithfully replicate the DNA. And so if we have cell A, which has a particular phenotype, and if we divide cell A, you agree, Stephen, that we got to make two cell A's. And they got to have exactly the same genes. And there can't be many changes. We've talked about that. And so not only the cell has to divide into two, but that means the genome has to divide into two. And furthermore, it happens because the cell divides. This is a bacterial cell. The chromosome divides. And as Daisy and Kelly and Aicha have told you, you have these little colonies that are clones of each other. That's the basis of genetic engineering. Remember, you don't clone genes. You clone cells with extra genes in them. And so we have these bacterial colonies as a result of cell division. So the property of the DNA specifies that the DNA has to replicate and it has to be stable along with some other things. So how does this happen? Well, clearly you saw in the movie last week that the double helix immediately told Watson and Crick how genetic information is passed on from generation to generation because of the fact that the double helix consists of two complementary strands. And Crick, you know, you saw in the movie, he orchestrated, you read in the book, that it was clear if the strands are complementary to each other, one strand specifies the other one specifies the other, and we can then get two identical strands after the cell divides. And so how this works is that one of the requirements for DNA replication has to be that you have a template. Don't ever forget that. You have to have a template that tells the cell what the sequences are of the new DNA molecules after DNA replicates because of complementary pairing. And notice that when that DNA replicates, you get two new molecules, two new daughter molecules. And notice that there's one old strand and one new strand. Meaning, here's a template. You get the formation of a new complementary strand. Here's the other strand, a template. You get the formation of a new complementary strand. Notice 5 to 3, 3 to 5, 5 to 3, 3 to 5. We have two double helices that are formed. That is called, conceptually, semi-conservative replication. And that's the process of DNA replication in bacteria and in humans. And it's called semi-conservative because in the new daughter molecules, those daughter molecules have one old strand and one newly synthesized strand. So the template strands are semi-conserved from generation to generation. It's also true, Lauren, that your DNA is a direct descendant of your ancestors going back millions of years. All of us, the DNA never dies, it's immortal, as was said by the actor who played Crick in the movie. It never dies. It goes back to Adam and Eve over and over and everything in between because of the replication of these DNA and the ability for it to pass on from generation to generation. And so here it is, DNA replication occurs semi-conservatively. And that's because of the complementary nature of the double helix, and I'm not going to go into it in any more depth, just about because you understand the double helix. Now notice here what I was telling you last week, that the DNA synthesis has to occur with certain polarity. Remember that. And remember that because the polarity of the DNA has 5 to 3 and 3 to 5, and remember the polarity of the sugar. The sugar was the hub, remember? 5' carbon has the phosphates. The 3' carbon has this little OH. And so the nucleotides, which are here, are brought to the replicating DNA by this enzyme DNA polymerase that we've talked a little bit about. So the DNA polymerase can recognize the nucleotides and recognize the template, which is one of the strands of the DNA molecule. And notice that this C here recognizes that there should be a G. This enzyme will bind where this little red rectangle is, and it will grab this nucleotide. It will allow this nucleotide to recognize that G. It will hydrogen bond, make a complementary base pair, and then that enzyme will make a 5', 3' phosphodiester bond and knock out these phosphates. And we have another nucleotide added to the chain, and it goes on and on and on. But notice that it starts at the 5' end with that phosphate, and it moves in the direction of the 3' end. RNA, as I just showed you, is always made 5' to 3'. DNA is also made 5' to 3'. And that's because, as I said, it has to have a polarity of sequence. And the polarity of sequence goes 5 to 3 always. And it's inherent in the transcription process, and it's inherent in the replication process. And as you can see here, conceptually, just conceptually, you can see a new phosphodiester bond. We have one more nucleotide added to this chain, and then another nucleotide will come along, so on and so forth. So we'll maintain the same order, and we'll maintain, more importantly, the same sequence because the template has a complementary base relationship. So there's the replication of the DNA. It's maintained for generation to generation. Now, in order to do DNA synthesis, you need certain things. And I want you to listen carefully. So I want you to really listen to every word that I have to say because it's very important for something that I'm going to talk about in 5 minutes. Notice that when you replicate the DNA, you need certain tools, to use an analogy back to your discussion. of course, you need the template, which is the mother DNA double helix. You need the DNA in the chromosomes. That's the ultimate template. The DNA in your chromosomes, the DNA in an E. coli chromosome, DNA in a viral chromosome, whatever. That's the template because it's passed on from generation to generation. So that, when I say template, provides the sequence information for the daughter DNA molecules. But then you need other things, one of which is a little tricky. You obviously need nucleotides because you can't make a DNA without nucleotides. You obviously need DNA polymerase because that's the enzyme, as Ben knows, that's the enzyme that recognizes the template and recognizes the nucleotides. Remember, enzymes catalyze reactions. DNA polymerase catalyzes the production of a phosphodiester bond. It does it by grabbing the template, grabbing the nucleotides, putting them and holding them together in the right combination like a glove in a hand, just as these green things are doing, so that the phosphodiester bond can be made. And so we need the template, we need the polymerase, we need nucleotides, and this little thing called the primer. And what is the primer? Notice here that we have a template, and notice we have this little tiny short sequence that's called the primer. The reason why we need the primer is that DNA polymerase alone, and I'm going to show you why in a second, DNA polymerase alone cannot initiate DNA replication. Are we all in agreement that cells have to replicate at specific times? They don't just do it all the time. When you have cancer, they do it all the time. We don't want that to happen. DNA replication has to be controlled. And one of the things, Anna, that helps it be controlled are these primers, because the polymerase just can't bind to the DNA willy-nilly and start replicating. It would be a disaster. Glioblastomas, like Senator Kennedy has, that is brain cancers, tragically are 100% lethal. There's absolutely no cure. And the reason why that glioblastoma is a glioblastoma is the brain cells are uncontrollably dividing because the DNA is uncontrollably replicating, because generally the cells don't divide and the DNA doesn't replicate. And so the division process and the replication process, Tom, has to be regulated. And one of the reasons why is because of these primers. In order for the polymerase to start replicating, it has to recognize the beginning of where replication occurs, just like a gene. Only this is sort of the beginning of replication for a whole chromosome. Albert, do you think there's a specific sequence there, or am I losing you? I can't hear you.
I think there's a specific sequence.
You think that sequence is the same as the promoter? It's DNA replication. Or do you think it's unique? What do you think? Is DNA replication the same as transcription?
No.
You sure?
No.
You aren't? Is replication of DNA the same as transcription in making a protein?
No, it's not.
Yeah, it's not. So would you think that the sequence that starts this process would be different than the sequence that is at the beginning of a gene? Because this has to be Yeah, exactly. So what do you think this little primer does then, Albert? What's your hypothesis? Where would it bind on the chromosome? Conceptually.
At the beginning?
Of?
The sequence and the chromosome.
That begins what? That initiates, that's responsible?
That initiates replication.
Yeah, so this little sequence binds to, what is the name of that sequence? You've had it in discussion and in your reading. It has a specific name. Come on, what is it? It's now third week. We've discussed it. I know you have in discussion. It's been in your handouts. What's it called? What's it called? Eric, what's it called? Yeah, it's called an origin of replication. So where do you think these primers bind, Brian? Yeah, they bind to the origin of replication. So the origin of replication has to have a specific primer. And that primer will bind to the origin of replication, and then the polymerase binds to the primer. That's why we have primers. Polymerase can't just bind by itself to DNA and allow it to replicate. So hold on. Okay, go ahead, Target. Microphone. You with me?
Is there only one primer per chromosome?
It gets a little tricky. Let me just try and keep conceptualizing. Generally, there's one primer. There could be more. Let's just say there's one primer sequence per kind of chromosome that's replicated, okay? And I'll show you what I mean by that in a second. The concept here is in order for DNA replication to occur, and you're going to see why I'm telling you this, is you need the template, you need the polymerase, you need the nucleotides, and you need this primer. Because the primer helps in the initiation of DNA replication, and it helps in the initiation of DNA replication because it recognizes a specific sequence, and that sequence is the origin of replication. Okay. Chris. If I told you that this process could occur in a test tube
Yes.
with any piece of DNA, if I could cause this process to occur in my lab with any piece of DNA I wanted, what would I need to cause that DNA to be copied? What are the things that I would need?
Well,
Just read them off.
the template DNA,
You need the template, which is the DNA I'm trying to copy.
the primer,
You need the primer. What would the primer be complementary to? There's no origin or replication. What would it be complementary to get this thing going? Might it be complementary to the piece of DNA I'm trying to copy?
yes,
Absolutely.
ideally.
So if you did this in a test tube, and you're going to see where I'm going in 3 minutes, you need the template, you need the nucleotides, you need the enzyme, and you need the primer. How do you get the sequence of a primer? Tell me, experimentally, how do I know what the sequence of the origin or replication is? How do I know what the sequence I should use for the primer should be, Chris? Did I have to clone the gene? Did I have to sequence the gene? Yes or no?
Sequence it, yes.
Yeah, you can't do this without knowing. You know the sequence of the origin, Max, by sequencing the origin and isolating it by recombinant DNA, just as you did the first week of class. You know the sequence of the primer to use to do this in a test tube with any piece of DNA because you've cloned the gene initially, you've sequenced it, and then you know what primer you can use. Hold off. You're doing great. Anyways, those are the requirements for doing DNA replication. And so, as I said, you need an origin of replication, which you've had in discussion. There it is. There's the origin of replication. There's the DNA polymerase, kind of crude. It's my drawing, but it binds to this particular origin, and it initiates the replication of DNA because it is a specific sequence, and the primers bind to that sequence, and the polymerase bind to the template and the primer. A DNA polymerase cannot initiate DNA synthesis under any circumstances without having the primer bound to the DNA, ever. Remember in discussion last week, Ben, we talked about what enzymes grab? In the case of DNA polymerase, the substrate for it is the template bound to the primer. Not simply a single-stranded DNA, but a single-stranded DNA with that little piece bound to it. That's the thing the polymerase recognizes. And you'll see why I'm telling you this in a second. The origin of replication in the case of E. coli is one unique sequence, Tiger. The chromosome of E. coli has an origin of replication. There it is, right here. It is a very specific sequence because the primers and all the other things, this is highly simplified, of course, have to bind to that particular point. Okay? Human chromosomes have a different origin of replication. Plant chromosomes have another origin of replication. When you clone something into those genomes, the foreign DNA is using the foreign origin of replication. That should be clear from using plasmids. It has its own origin of replication. And in the case of E. coli and humans, DNA replication starts with the origin of replication and goes in two different directions. It's bidirectional. Catherine, what's your hypothesis for why something will start at the origin and move out bidirectionally, rather than just going around in the same direction?
It's faster.
Exactly. And why does it have to be fast? What's happening at the same time the DNA is replicating? where is this going on? Outside or inside a cell?
Inside?
Yeah. Does a cell have a certain time in order to divide?
Yes.
What would happen if the DNA took longer than the cell did to divide? What would happen?
Would it end up without DNA?
Yeah, it'd be dead, wouldn't it? So there's a race. This has to be timed. And if the cell divides fast, the DNA's got to divide fast. And in the case of E. coli and other bacteria, the only way you can accomplish this, remember, polymerase is a machine. It's a little nano machine. It's moving like a locomotive along the DNA. It only can move so fast. And because it's a certain size, you know, in E. coli, 5 times 10 to the 6 nucleotides, because the cell divides in a certain time, pretty quickly, minutes, it's got to go in 2 directions. And that's what happens. It happens in humans too, but I'm not going to get into that. So here's our origin of replication. Notice that we're looking at an electron micrograph. Notice that there's a bubble here. Here's the template. And here's the 2 new strands that are forming. And notice they get bigger and bigger and bigger. And eventually there'll be 2 completely separate chromosomes. There'll be 2 separate chromosomes. Much like that at the end of this process. So we have the origin of replication, and we have DNA, and there it is again. Origin of replication is right here, right here. It's going out. DNA replication is going in this direction. DNA is going in this direction. Here's one daughter double helix. Here's another daughter double helix. Remember, it has 1 template strand, 1 newly synthesized strand, and it's simply the most beautiful process that you can ever imagine. Tiger, here's your sequence. This is the sequence of the origin of replication of E. coli. Notice that it's a very specific sequence here. It consists of this sequence and that sequence. It's a little more complicated than I told you, but it is a specific sequence. This is where the primers bind, and this is where all of these different proteins bind, and those proteins have to bind because they're involved in telling the polymerase when replication occurs. And that's highly regulated. Regulates in the case of bacteria what environment you're in, the conditions, all these other kinds of things. This is a highly regulated process both in humans and in bacteria.